Good morning, dear student. How are you today? Hope you are fine and safe all the time. Today, in this lecture, we are going to illustrate the mechanism of translation. As we dealt in the previous lecture, the tools of translation. Now, we are going to illustrate how translation takes place. Translation is the process by which a protein is synthesized from the information contained in a molecule of the messenger RNA or the mRNA. During translation, an mRNA sequence is read using the genetic code, which is a set of rules that defines how mRNA sequence to be translated into the 20-letter code of amino acid, which are the building block of proteins. The genetic code is a set of a 3-letter combination of a nucleotide called codon, each of which correspond with a specific amino acid or a stop signal. Translation occurs in a structure called the ribosome, the place where translation takes place, which is a factory for the synthesis of the protein ومصنع تصنيع البروتين. The ribosome has a small subunit and also a large subunit, okay? Composed of ribosomal RNA or rRNA and a number of proteins. Here we have the mRNA that will be translated into the amino acid. As you see here, we have a growing chain of amino acid. Now we are going to see a video with each other about the process of transcription with the translation. Okay, let's start in order to know the mechanism of translation.
After learning about DNA, have you ever wondered how can the DNA actually result in a trait? Let's take an example, like eye color. Yes, your DNA has the genetic information that codes for the color of your eyes. Your eye color is based on a pigment that is inside the eyes. But in order to have that pigment, you have genes.
So عم بقلنا إنه DNA code for the eye color. You know that the eye color هي صبغة pigment. ممكن يكون اللون العيون خضراء زرقاء بني. هود البيجمنت يعني لون العيون جايين من أمر من جين. هيدا الجين الموجود طبعا بالDNA.
Are portions of DNA that can code for proteins which help make that pigment. So what we're going to talk about is how your DNA can lead to the making of a protein. This process is called protein synthesis. Synthesis essentially means to make something. So protein synthesis means to make protein. And you may wonder, what's a big deal about proteins? Well, you may not realize this, but proteins are kind of a big deal. They do all kinds of things. Proteins are involved in transport, in structure, in acting as enzymes that make all kinds of materials, in protecting the body, and so much more.
So عم بقلنا هون البروتين ممكن يكون انزيم، ممكن يكون هرمون، ممكن يكون antibody in order هيدا الانتيبادي in order to protect the body، ممكن يكون بيعمل structure. يعني أكيد نحنا اللحم يلي بجسمنا العضلات هيدي made up of protein. okay هيدا كل الفانكشن في عنا many function of the protein.
You've got to make proteins. It's essential for you to live. And what is so cool is that you are making proteins right now as you sit and watch this video. It's happening in your cells. They're making proteins. So back to your DNA and its role in all of this. All of your cells have DNA. Well, a few exceptions. And that DNA is in the nucleus. Some DNA
So the DNA contains the nucleus, okay?
Is non-coding DNA. Some DNA makes up genes that are not activated. More about that in our gene regulation video, but we're going to talk about genes that are coding for active proteins. So how are we going to get the information from these genes out of the nucleus so that the cell can start?
So يا مس بالترانسليشن هيدول المعلومات يلي موجودة بالدي ان اي you know that هيدي المعلومات بتروح على من النيوكليوس into the cytoplasm by the mRNA يعني هيدي
producing the proteins that
عم تشرح هلا هي عن الترانسكريبشن
needs to make.
يعني شي نحن
Well, let us introduce you to the amazing work of RNA. We have a video comparing and contrasting RNA and DNA. We'll be brief here in saying that RNA is a nucleic acid, like DNA.
DNA هي double helix والRNA it's a single helix. هيدي nucleic acid also هيدي nucleic acid. Okay?
But it has a few differences. Its role in protein synthesis also is huge. Before we get into the process, please note our typical disclaimer. We tend to simplify topics, but as always, we hope that you have the desire to explore this complex, amazing process later on, to learn all about the extra information that we don't have the ability to include in this short video. In protein synthesis, we can look at two major steps. One is transcription, and the other is translation. Transcription has a C in it, and translation has an L in it. I remember that C comes before L in the
هون مس عم بتقلنا إنه فزلكي يعني شوي بالحكي. transcription طبعاً أول شي بعدين transcription تصنيع mRNA translation تصنيع البروتين starting from the mRNA. عم بتقلنا هون لأنه السي قبل ال بالألفابت بالABC فتذكروا إنه transcription قبل translation. Okay يعني not important
alphabet, which helps me remember that transcription comes first. I like a lot of alphabet mnemonics. Now, transcription is when we're going to transcribe the DNA into a message. In your cells, the DNA is in the nucleus, so therefore we're doing transcription in the nucleus. In the step of transcription, an enzyme called RNA polymerase will connect complementary RNA bases to the DNA. These RNA bases are bonded together to form a single-stranded mRNA. The M in mRNA stands for messenger. Messenger RNA consists of a message made of RNA that has been based on the DNA. We do want to mention that this mRNA is not usually ready to go right away. There's usually a significant amount of mRNA editing that occurs. We highly encourage you to do some reading about that because it's not only fascinating, it's critical for the process to work correctly. So what's something great about being mRNA? Well, in eukaryotes, you get out of the nucleus. The mRNA can go out of the nucleus into the cytoplasm where it's going to attach to a ribosome. Ribosomes make protein. The ribosome is made of rRNA, and that's an easy one to remember because the R stands for ribosomal RNA. The ribosome is going to build our protein in the next step called translation. You know, you can find a lot of great clips and animations on translation that are just fantastic. We're just going to break down some basics of what's happening. In the cytoplasm, if you look at this, you have all these tRNA molecules available. tRNA stands for transfer RNA. They carry an amino acid on them. An amino acid is the monomer for a protein. It's a building block for protein. Since we're making proteins, we're going to need those amino acids to build it. If you have a bunch of amino acids together, you can build a protein. So it's the tRNA that is going to bring those amino acids together to make that. But wait, how does the tRNA know which amino acids to bring? That's why the mRNA, the message, is so important because it's going to direct which tRNAs come in and therefore which amino acids are transferred. All of these tRNAs are looking for complementary bases. When they find the complementary bases on
بس هيدي mRNA اللي اجت على على الريبوزوم in order to be translated into a chain of amino acid. هيدي transfer RNA اللي نحن قلنا هي translator يلي بتفهم باللغتين لغة الامينو acid and لغة النيوكليوتايد. اوكي فبتيجي هيدي يمس اللي هون موجود الانتي كودون complementary لهيدا الكودون يلي هون.
mRNA, they transfer their amino acid. When tRNA is bringing in the amino acids, it reads the bases, represented by these letters here on the mRNA. in threes. So it doesn't read one letter at a time, it reads in triplets. That's called a codon. So for example, in this mRNA, the tRNA
يعني عم بتقلنا يا مس ما بتقرا هي بس 1 نيوكليوتايد أو 2 بتقرا تريبلت يعني A U G. شو الكومبلمنتري لل A U G؟ It's UAC. هاي الكومبلمنتري اللي إلها بتيجي بتقعد هون، يعني هي بتقرا 3 هودي الثلاثة اسمهم triplets and also called a codon. هيدول codon هون يلي بالتي ار ان اي يلي هون اسمهم anticodon which are complementary to
it
the
would
codon.
read the codon AUG. One of these tRNAs contains a
anticodon.
complementary anticodon, which in this case is UAC. All tRNAs that have the anticodon UAC will be carried.
دايما الترانسكريبشن بدها تبلش الترانسليشن بدها تبلش بهيدا التريبلت الاي يو جي. الاي يو جي اللي هو الانتي اللي او الكومبلمنتري اللي له يو اي سي. اليو اي سي مترجم يا مس معناته ميث ايونين. اي بروتين يا مس لازم يبلش to start with this amino acid that is called methionine. Okay?
An amino acid called methionine. A tRNA with the UAC anticodon comes in to pair with the complementary AUG codon on the mRNA. It transfers the amino acid it carries, methionine. The tRNA will eventually leave, but it will leave behind its amino acid.
شفتوا كيف يا مس؟ قعد الأول هيدا التي ار ان اي قعد على المسنجر ار ان اي باي ذا لأنه هو كومبلمنتري للنيوكليوتايد هير، وكان حامل من فوق ميثايونين صح اللي هو الفيرست امينو اسيد. برجع لما قعد الثاني عمل بيبتايد بوند بينه وبين هيدا الامينو اسيد اللي هون، عمل بيبتايد بوند وراح هيدا الترانسفير ار ان اي مش بعده موجود لانه انا ما بيهمني هيدا الضل، المهم انا اعمل chain of amino acid. Okay, in order
it's the first amino acid before looking at the next codon. Before we do the next codon to carry this on, if you're wondering, how did you know that the tRNA that went with the AUG codon would be carrying an amino acid called methionine? Well, for that, you will find a codon chart helpful.
نحن قلنا انه بالجينيتيك كود تيبل هو الديكشنري لحتى انا ترجم من لغة النيوكليوتايد للغة الامينو اسيد. سو الميثايونين يا تيتشر وينه مش موجود هون الميثايونين.
Learn to use a codon chart to determine which amino acid each mRNA codon will code for. Isn't it so fascinating that scientists have been able to determine which amino acid corresponds with these codons? I used to have a codon chart poster and just marvel at that. You could see on a codon chart that the AUG codon on the mRNA codes for methionine.
ما كنت شايفيتها يا مس. ال AU هيك نحن قلنا بنقراها AUG، هالأول هالتاني وهالتالت code for methionine. so ال AUG الأنتي كودون اللي إلها بيكون UAC حامل الترانسفير RNA بتكون حاملة الميثايونين. okay?
AUG is also considered a start codon, as methionine is typically going to be your first amino acid in proteins. There are many types of amino acids in the codon chart, but there are even more possible codon combinations. That means there can be more than one codon that code for the same amino acid. For example, according to the mRNA codon chart, all of these mRNA codons here code for the same amino acid, leucine. That means their complementary tRNA.
شايفين يا مس؟ هيدول ال UAA وال UAG وال CUU وال CUC وال CUA وال CUG كلهم code for leucine. هيدول اللي أنا سميتهم they are called redundant triplet or redundant codon. ماشي. يعني شايفين هيدي ال AAC حاملة ليوسين، ال GAA ليوسين، ال GAC ليوسين، كلهم حاملين ليوسين لأنه في عنا كذا واحد code for ليوسين. Okay?
These all carry the same amino acid, leucine. Okay, so going back to the mRNA, let's try the next codon on this mRNA, CCA. On the codon chart, you can see that codes for the amino acid proline. The complementary tRNA has the anticodon GGU. And look, there's the proline that we knew it would be carrying. The tRNA will transfer that amino acid and eventually leave where it can pick up another amino acid. These amino acids are held together by a peptide bond, and it will keep on growing. Typically, at the very end of the mRNA, there is a stop codon. Stop codons.
So هيدا يا مس they are UAG هيدا stop codon، يعني بتيجي لعنده ال mRNA وبتوقف تصنيع أو إنه تمد هيدي ال chain of amino acid لأنه هو أمر إنه خلص stop.
Not code for an amino acid, but when the ribosome reaches it, it indicates that the protein.
How many stop codon do we have? UAA, UAG, and UGA. كلهم هودي stop codon. Okay? It doesn't code for amino acid, but it code for stop the growing chain of amino acid.
Building is finished. So the result of translation is that you built a chain of amino acids that were brought in certain sequences based on the coding of the mRNA. But remember, that mRNA was complementary to the DNA, so the DNA ultimately was the director of the entire protein building. Of course, it couldn't have done it without some serious help from mRNA, rRNA, and tRNA. Protein folding and modification may occur, and the protein may need to be transported. This can all vary based on
folding لهيدا البروتين ليتحول ل 3D structure.
Protein structure and function.
In order to be functional.
Another fascinating topic for another Amoeba Sisters video. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
بس انتو شفتوا بالفيديو هلا مثل ما شفنا كيف بيصير في عنا ال growing chain of amino acid. بدي منكم كل الستبس يلي شفناهم بالفيديو موجودين هون. Translation steps أول شي مكونة من 3 stages: initiation, elongation, termination. initiation مطرح ما بتبلش الترانسليشن اللي هو start codon اللي هو الميثايونين. The elongation and the elongation يعني the growing chain of amino acid, but the termination لما توصل للstop codon. طبعا هودي مطلوبين مننا للحفظ ولازم نكتبون على الدفتر، وأي حدا عنده question أو ما كان واضح عنده عملية الترانسليشن you have to ask me on my private. Okay? Thank you for watching.